So far, we've transcribed and we've processed the messenger RNA in order to form a mature messenger RNA molecule. The next stage in the game will be to move on to the translation process. But before we do, we need to understand a little bit about transfer RNA and ribosomes. Now, transfer RNA molecules are molecules that carry amino acids to the ribosome. We've already learned that the ribosome is the site of protein synthesis, but how do we get the amino acids there? This is where tRNA comes in. Here's a tRNA molecule over here. It has an anticodon end, which has a series of nucleotides that match the codon on the messenger RNA. And it has an acceptor end to which we attach a specific amino acid. The enzyme that does this is called aminoacyl-tRNA synthetase. There's an aminoacyl tRNA synthetase specific to each amino acid. Here's an image of the enzyme below. Aminoacyl tRNA synthetase is specific to an anticodon on a specific tRNA. In this case, we're looking at the tRNA that carries tryptophan. However, it's not carrying it at the moment. We need to load that tRNA. The tRNA goes into the binding site on the enzyme, and then the enzyme attaches tryptophan to its acceptor end, and then releases the charged tRNA. That is the tRNA that's now carrying the amino acid tryptophan. When we go back to the building site analogy, I think of the tRNAs like the trucks that are going to the building site. The ribosome is the building site. The messenger RNA is a copy of the blueprint. And the amino acids are the bricks that we're building our new structure with. So again, we'll have codons on the messenger RNA that will match up with the anticodons on the tRNA in order to allow specific amino acids to be dropped off at the ribosome to generate a polypeptide chain. So each aminoacyl tRNA synthetase recognizes only one amino acid, but several tRNAs because of the redundancy in the genetic code. Remember, sometimes several codons may code for one specific amino acid. When a tRNA is charged, it has an amino acid added using the energy from ATP. This ATP is utilized to form peptide bonds without any additional energy. That's why we call the tRNAs charged. Not only do they carry the amino acid, but they carry the energy necessary to allow the formation of peptide bonds between amino acids in the growing polypeptide chain. It's really important that the aminoacyl tRNA synthetases do their job because the ribosomes don't verify whether they're bringing in the right amino acid or not. Let's watch a quick animation that might help us clear up how the tRNA charging works.
Aminoacyl-tRNA synthetases catalyze the formation of charged transfer RNA. To begin the process, a specific aminoacyl-tRNA synthetase binds a particular amino acid and also a molecule of ATP at its active site. A covalent bond is formed between AMP and the carboxyl group of the amino acid, and pyrophosphate is released from the enzyme. A specific tRNA that has an anticodon that corresponds to the amino acid then binds to the synthetase. The RNA sequence in the anticodon region, as well as other parts of the tRNA molecule, such as the acceptor stem, are important for recognition between the tRNA and the aminoacyl-tRNA synthetase. The bond is broken between the amino acid and AMP, and the AMP is released. At the same time, a covalent bond is formed between the amino acid and the 3' end of the tRNA, which is now called a charged tRNA molecule. The charged tRNA is then released from the enzyme and is now ready to be used in the process of translation on a ribosome.
Now let's look closer at the ribosomes. Ribosomes have multiple tRNA binding sites. There's a P site, an A site, and an E site. A ribosome is composed of a small subunit and a large subunit. I like to think of it as much like a hamburger. It's going to clamp down on the messenger RNA in order to translate it from RNA language into polypeptide language of amino acids. The P site binds the tRNA that's attached to the growing polypeptide chain. The A site binds the tRNA that's carrying the next amino acid. It's sort of like the waiting room. And then the E site binds the tRNA that carried the last amino acid. So as the ribosome moves along the messenger RNA molecule from the 5' to the 3' end, because ribosomes are a little bit different. They move beginning here at the 5' GTP cap. They move along the messenger RNA in this direction. So first, an amino acid will arrive in the A site, or the arrival site. As the ribosome moves along one spot, that one will move into the P site, and a new one will arrive in the A site. As it moves along one spot again, the tRNA from the P site will end up in the E site, or the exit site. We'll see how this goes into motion shortly. The ribosome has two primary functions. First of all, to decode the messenger RNA, and then to form peptide bonds between each of the amino acids that are delivered to the ribosome. The enzyme responsible for forming the peptide bonds is called peptidyl transferase, and that's because it transfers the peptide chain from one site to the next site. It's the enzymatic component of the ribosome. Translation is also broken into three phases called initiation, elongation, and termination. Again, if we think about logically what has to happen, you understand what occurs in each of these different phases. Initiation is where we get everything together. We're going to assemble the ribosome with the messenger RNA. In elongation, we'll be elongating the polypeptide chain by adding amino acids. And during the termination phase, we'll reach a stop codon, which codes for release factor and ends the process of translation. As I've previously introduced, translation is much like building a house, where the protein is the house. The amino acids are the bricks. The tRNAs are the trucks bringing bricks to the building site. They'll go back and refill and pick up new bricks at the aminoacyl-tRNA synthesase loading stations. And then messenger RNA is a copy of the blueprint giving instructions of which bricks to put down where. So let's look at the initiation. In prokaryotes, the initiation complex includes an initiator tRNA that's charged with N-formyl methionine. Just remember that it's methionine because the start codon is the same for every single amino acid chain. The small ribosomal subunit arrives first. The first tRNA with an initiation factor will land itself in what will become the P site of the ribosome. And then the messenger RNA strand will slide in there beneath it, matching up with the anticodons on the first methionine tRNA. It's only after this has happened that the large subunit assembles itself over the tRNA and the messenger RNA. We now have one actively charged tRNA carrying methionine in the P site. We have an empty A site and an empty E site. Initiation in eukaryotes is really similar, except the initiating amino acid is methionine. This is the one you want to remember. It's a more complicated initiation complex, just meaning that it involves many more proteins. And they also lack the ribosome binding sequence because eukaryotic mature messenger RNA has a 5' GTP cap, and the small subunit binds directly with that. Let's look in the elongation phase. Elongation is where we add amino acids. The second tRNA will bind in this empty A site or arrival site. And here, a peptide bond will form between the amino acid of the tRNA in the P site and the amino acid number 2 that's now in the A site. Both these amino acids will now end up on the A site tRNA. This cycle of addition will continue as we grow the new polypeptide strand. The next amino acid will end up in the A site, steal the chain from the P site, so on and so forth. Let's look at this in a little bit more detail. First, we'll have a charged tRNA arrive in the A site. This charged tRNA has the energy and an elongation factor to allow the transition of the polypeptide chain in the P site over to the amino acid that's being held in the A site. So here we have the tRNA in the P site. It's now able to transfer its amino acids over to the one in the A site. The ribosome translocates towards the 3' end by one codon, leaving the A site empty, and the E site ejects the empty tRNA, which can now go around and pick up another amino acid at the amino acid tRNA synthetase loading stations. Again, GTP charged with elongation factor tRNA arrives in the A site. It drops off its energy in the elongation factor, which is now going to allow peptide bond formation. The P site tRNA transfers the chain over into the A site, leaving itself empty so that as the ribosome translocates one codon, it can drop off the empty, ejected tRNA and leave a space for the arrival of a new tRNA. Let's watch a quick animation of this. After all, moving pictures are worth way more than 1,000 words, and this one really warrants some clarification.
Once an initiator methionine tRNA complex is positioned at the AUG start codon, elongation of the amino acid chain proceeds. The next aminoacyl tRNA binds the ribosome at the A site. The amino acid at the P site is transferred to the amino acid attached to the tRNA at the A site. The amino acids are linked by the formation of a peptide bond. Peptide bonds are formed by reaction of the alpha amino group of the A site amino acid with the carboxyl group of the P site amino acid. After peptide linkage, the empty P site tRNA shifts to an exit, or E site, on the ribosome. The ribosome translocates the length of one codon, releasing the empty tRNA. The A site is now free for the next incoming tRNA. The cycle is repeated as the ribosome travels along the mRNA, resulting in a growing polypeptide chain.
So here again is the elongation cycle. Spend some time in here. See if you can diagram which piece moved where. Arrival in the A site. P is the polypeptide site. E is the exit or the ejected site. Work through this cycle and understand it well enough to be able to diagram and explain. Now, there are fewer tRNAs than there are codons. That is because one amino acid may be coded for by several different codons, and we only need one tRNA for each amino acid. Because of this, we'll see some wobble pairing. It allows less stringent pairing between the 3' base of the codon and the 5' base of the anticodon. So the wobble occurs usually in the last digit as we load amino acids onto specific tRNAs. Because of this wobble, we can afford to use fewer different tRNAs to accommodate all of the different codons. Finally, we get to the termination of translation. Elongation will continue until the ribosome encounters a stop codon. Once it encounters a stop codon, a release factor will come in. A release factor is not a tRNA. It's distinctly different. In fact, I think of it as a bomb. Once the stop codon codes for a release factor, Essentially, it blows up the whole thing. The ribosome is released, the messenger RNA is released, and the new polypeptide chain is released. In eukaryotes, translation may occur in the cytoplasm or in the rough endoplasmic reticulum. If this happens, there are signal sequences at the beginning of the polypeptide sequence that bind to a signal recognition particle on the rough endoplasmic reticulum. The signal sequence and SRP are recognized by the rough ER receptor proteins, and the docking holds the ribosome to the rough ER. As the polypeptide chain is formed, it's dumped inside the rough endoplasmic reticulum. This is the beginning of the protein trafficking pathway that you learned about when we considered cell organelles and the endomembrane system. In this table, we can summarize some of the differences between prokaryotic and eukaryotic gene expression. Spend a little time looking at it just to see if you understand the distinctions. Let's take a quick card quiz to finish up this section. Aminoacyl-tRNA synthetases do which of the following? Empty tRNA molecules leave the ribosome at the start codon, AUG, also codes for methionine. How does this affect the protein? All stop codons code for proline. Thus, proteins begin with methionine and end with proline. Is this true or false? Peptidyl transferase forms peptide bonds. Where is this enzyme located? Where does translation take place? Here are your card quiz answers. Please take some time getting to understand how the process of translation works. There are plenty of animations out on Connect, and then spend plenty of time in LearnSmart.